Somewhere in a climate-controlled vault in Jerusalem, a piece of animal skin no bigger than a postage stamp has been sitting under glass for decades, studied letter by some of the sharpest minds in biblical scholarship. Everyone was looking at what was written on it. Nobody thought to ask what it was written on. That single oversight held for 70 years, until a scientist who studies microscopic worms sat down next to a Bible scholar on a shuttle bus and asked one quiet question that would eventually unravel everything the world thought it understood about the most famous ancient texts on Earth. What that question uncovered is still rewriting history right now in real time, and the story is far from finished. For nearly 7 decades, the Dead Sea Scrolls held their secrets in plain sight, and almost everyone who studied them was asking the wrong question. The scrolls themselves are not a mystery in the sense of being hidden or lost. Since 1947, scholars have had direct access to roughly 25,000 fragments of ancient parchment and papyrus pulled from 11 caves near the ruins of Qumran on the shores of the Dead Sea, representing close to 1,000 separate manuscripts written between the 3rd century before the Common Era and the 1st century after it. These fragments contain the oldest known copies of Genesis, Isaiah, Psalms, and dozens of other biblical and sectarian texts. Documents that had quietly survived the fall of the Second Temple, the collapse of the Roman Empire, and every political upheaval that followed for 2,000 years. And yet, for all that access, for all that scrutiny, the scrolls refused to give up an easy story. The question that consumed generations of researchers was simple to ask and brutally difficult to answer. Who wrote these, and why were they hidden here? The discovery itself began with an accident so small it barely registers as the beginning of anything significant. In 1947, a young Bedouin shepherd named Mohammed ed-Dib was searching the hills above the Dead Sea for a goat that had wandered off from his flock. Coming across the mouth of a cave, he tossed a stone inside, more out of curiosity than purpose, hoping the sound might startle the missing animal into view. Instead, he heard something shatter. What he found when he climbed inside were ancient clay jars, sealed for centuries, containing leather scrolls wrapped carefully in linen cloth. He had no way of understanding what he had just found, neither at first did anyone else. But over the following 9 years, as word spread and archaeologists descended on the region, cave after cave near Qumran yielded more fragments, more jars, more pieces of a puzzle nobody yet understood the shape of. This was not one dramatic discovery. It was a slow, accumulating flood of ancient material that would eventually become one of the most significant archaeological finds of the 20th century. Once scholars had the fragments in hand, the real work began, and for most of the next 70 years, that work relied entirely on the human eye and human judgment. Researchers compared handwriting stroke by stroke, trying to determine which fragments had been written by the same scribe. They studied the color, texture, and thickness of the parchment itself, hoping visual similarities might reveal which scattered pieces had once belonged to the same original scroll. They cross-referenced theology, calendar systems, and literary style, searching for patterns that might connect one fragment to another across thousands of disconnected pieces. And from all of that painstaking, genuinely brilliant scholarship, a theory emerged that would dominate the field for generations. The scrolls, most experts came to believe, belonged to a Jewish sect called the Essenes, a secluded ascetic community described by ancient historians like Josephus as devoted to purity, scripture copying, and preparation for a coming divine judgment. The ruins at Qumran, sitting just beside the caves, looked exactly like the kind of settlement such a community might have built. The pieces seemed to fit almost too neatly. Case closed, or so it seemed for decades. But underneath that tidy explanation, uncomfortable details kept quietly resurfacing, details that the tools available at the time simply could not resolve. The scrolls showed a strange and often bewildering degree of textual variation. Different versions of the exact same biblical book sat side by side within the same collection, written in different scripts, by scribes of clearly different skill levels, on parchment of wildly inconsistent quality. When radiocarbon dating was finally applied to a sample of the fragments in the early 1990s, some of them turned out to predate the Qumran settlement itself, a chronological contradiction nobody could fully explain within the existing framework. And there was a deeper, more unsettling question sitting beneath all of it that the Essene theory never adequately answered. Why would one small, tightly disciplined religious community, supposedly built around strict uniformity of belief and practice, hold onto multiple contradictory versions of its own sacred scripture? Why keep 2 entirely different editions of the book of Jeremiah, one substantially longer than the other, with whole passages missing and chapters, rearranged inside a community that was supposed to represent a single unified religious vision? Some researchers floated alternative explanations at the margins of the field, suggesting Qumran might have been a pottery workshop, a military outpost, or a sacred depository where worn-out texts from many different communities were respectfully retired rather than simply discarded. None of these ideas carried enough hard evidence to unseat a paradigm that had held firm for generations. The field had reached a wall, and everyone studying the scrolls knew it, even if nobody could see a way past it. Then, in 2012, something happened that had nothing to do with archaeology at all. At Tel Aviv University, a molecular biologist named Oded Rechavi, whose actual research focused on inheritance patterns in microscopic nematode worms, found himself sitting next to a biblical scholar named Noam Mizrahi during a new faculty orientation program. The two struck up a conversation, the kind of easy small talk that happens between colleagues meeting for the first time, and it eventually drifted toward Mizrahi's work on the Dead Sea Scrolls. Mizrahi laid out the core problem as plainly as he could. 25,000 fragments, no titles, no page numbers, no reliable way to determine which piece belonged with which beyond handwriting comparisons, and educated guesses about the age and texture of the leather. It was a system, he admitted, that was subjective, imprecise, and fully capable of being catastrophically wrong without anyone realizing it. Rechavi listened, and then he asked a question that nobody studying these scrolls had apparently thought to ask before. What exactly is the parchment made of? Mizrahi answered simply that it was animal skin. Rechavi paused, and then said something that would quietly change the direction of an entire academic field. Animal skin has DNA in it. That single observation opened a door nobody had walked through before. Rechavi specialized in ancient genetic analysis, a method that had already transformed the study of human prehistory, allowing scientists to trace migration patterns and population history across tens of thousands of years using nothing but degraded genetic material recovered from bone, teeth, and tissue. But nobody had ever thought to point that same toolkit at a manuscript, at scripture itself, treating the parchment not as a passive surface for holy text, but as a biological artifact in its own right, carrying its own separate history inside its molecular structure. Rechavi and Mizrahi began assembling a team, eventually joined by geneticist Matthias Jakobsen of Uppsala University in Sweden, genomics specialist Christopher Mason of Weill Cornell Medicine in New York, and Penina Shore, the curator, responsible for the Dead Sea Scrolls collection at the Israel Antiquities Authority. What began as a lunch conversation between two university colleagues would eventually stretch into a 7-year research effort, one that none of them could have fully anticipated the outcome of when it started. The first obstacle the team faced was serious enough to have stopped the project before it began. The Dead Sea Scrolls are sacred to multiple religious traditions and are, by any definition, entirely irreplaceable. Any method requiring cutting, drilling, or physically removing material from an inscribed fragment was completely off the table. So how exactly do you extract usable genetic information from an object you're absolutely forbidden to damage? The answer had been sitting there the entire time, hiding in plain sight inside the storage boxes themselves. Decades of careful handling had left behind fine particles of parchment dust, quietly accumulating at the bottom of archival containers. That dust contained collagen, the structural protein found throughout animal skin, and locked inside that collagen, preserved across 2,000 years, was ancient genetic material. By carefully collecting dust from blank margins and worn, unwritten edges, the researchers could gather enough material for analysis without ever touching a single inked letter of sacred text. The evidence had been sitting there quietly through every decade of scholarly debate, waiting for someone to think to look at it differently. Turning that dust into usable genetic sequences, however, was brutal, exacting work that took years to get right. The very process of making parchment in the ancient world, soaking animal hides, scraping them clean, stretching and drying them under tension, damages DNA at a molecular level from the moment of production. The iron-based inks used on many of the scrolls are chemically corrosive, further degrading any genetic material nearby. And after 2,000 years sitting in desert caves, followed by 70 years of handling by hundreds of researchers, curators, and visitors, the risk of modern contamination was constant and serious. The team worked inside dedicated ancient DNA facilities that more closely resembled surgical clean rooms than traditional archives, requiring full protective suits, positive air pressure systems, and ultraviolet sterilization to prevent even a single stray human skin cell from corrupting a sample. They built strict authentication protocols to confirm that any genetic signal they detected was genuinely ancient rather than modern contamination, searching specifically for patterns of molecular decay that could only accumulate gradually over centuries, and running every sample through multiple independent extractions, cross-checked between separate laboratories. It took years of repeated failure before the team finally assembled genetic sequences reliable enough to trust, drawn from 26 actual scroll fragments out of hundreds of samples processed. And when Rechavi finally sat down to study what those sequences revealed, the results were more disruptive than anyone on the team had expected. Most of what the DNA showed lined up comfortably with existing assumptions. The overwhelming majority of the parchment samples came from sheep, an animal fully capable of surviving in the harsh, arid environment surrounding Qumran, exactly what researchers would expect from a community producing scripture locally in the Judean desert. But a smaller cluster of fragments came back, showing something else entirely. something that immediately complicated the picture: cowhide. Mizrahi reportedly sat with that specific result for a long time before saying anything to his colleagues, because he understood immediately what it meant, and it was not a minor footnote to be quietly set aside. Qumran sits beside the Dead Sea in one of the driest, most punishing environments found anywhere on the planet. Sheep and goats are built for exactly that kind of terrain, able to survive on minimal water and tough scrub vegetation. Cattle cannot survive under those conditions. A single cow requires dozens of gallons of water daily, along with substantial grazing pasture, resources that simply did not exist at Qumran in any form that could have sustained livestock. There was no plausible version of a community living at Qumran that could also have been raising cattle nearby, which meant, quite simply, that any scroll fragment written on cowhide had to have originated somewhere else entirely, somewhere with real water and real grassland, and had then physically traveled to the caves where it was eventually found. The clearest and most striking example of what this meant in practice involved the Book of Jeremiah. Two dramatically different versions of this book survive among the scrolls. One closely resembles the Jeremiah familiar from modern Hebrew Bibles, longer, with its chapters following the expected traditional sequence. The other is noticeably shorter, missing entire sections outright, its remaining chapters arranged in a completely different order. Before 1947, the earliest available Hebrew manuscripts of the Old Testament dated only to roughly the year 1000 of the Common Era, and for decades after the scrolls were discovered, scholars had debated endlessly over what these two divergent versions of Jeremiah actually represented. Was one an early draft? Was the other a later theological revision? Was the difference simply the result of a careless scribal error compounding over generations of copying? The DNA evidence finally gave the field a concrete physical answer to a question that had been argued over for decades without resolution. The shorter version of Jeremiah was written on cowhide. The longer, more familiar version was written on sheepskin. Different animals, almost certainly sourced from entirely different regions. These were not simply two copies slowly drifting apart from one shared original text over time. They were two genuinely independent textual traditions, developed separately in two different communities in two different parts of the ancient world, and only later brought together in the same set of caves. The same genetic approach applied to a liturgical text called the Songs of the Sabbath Sacrifice, a work describing angelic worship inside a heavenly temple, produced an equally revealing result. Copies of this particular text had been found both at Qumran and at Masada, the mountaintop fortress where Jewish rebels made their final desperate stand against Rome in the year 73. Genetic analysis of the Qumran fragments showed that multiple copies of this same text had been made from entirely different, unrelated animals. In other words, this was not a guarded, exclusive text belonging to one isolated sect. It was popular. It was widely copied. It was independently produced in multiple locations across the region, circulating among communities that were clearly not confined to a single settlement beside the Dead Sea. Layered on top of this, the team's kinship analysis, which examined nuclear DNA markers capable of identifying animals that were closely related to one another, siblings or members of the same flock, revealed something equally significant. Certain clusters of fragments, clearly produced from a single flock around the same narrow window of time, turned up sitting alongside other fragments with no genetic relationship whatsoever, coming from entirely different animals and entirely different flocks. This was not a collection that had grown naturally out of a single central scriptorium. It had been gathered together from multiple points of origin, representing multiple communities, spanning multiple decades, and quite possibly stretching across entire centuries. Taken together, this evidence did not simply add a footnote to the Essene theory. It dismantled the foundation the theory had been built on. If the scrolls truly represented the private library of one isolated, disciplined sect living at Qumran, their sheer internal diversity makes very little sense. Why would a single community bother carefully preserving competing, contradictory versions of its own sacred scripture rather than simply discarding the ones it considered incorrect? But if the collection had instead been gathered gradually from many different Jewish communities, scattered across ancient Judea, and finally sealed away together during one catastrophic historical crisis, everything falls into place with far less strain. The diversity stops being a complication that needs explaining away and becomes, instead, the entire point of the collection. This genetic evidence backed up something a handful of scholars had long suspected, but had never been able to prove using textual analysis alone. The caves near Qumran likely functioned as a geniza, a sacred storage site where worn-out or superseded texts, considered too holy under Jewish law to simply destroy, were respectfully deposited by communities from across the surrounding region. Jerusalem sits only a short journey from Qumran by ancient standards, and when Roman forces laid siege to the city between the years 68 and 70, it is entirely plausible that Jews fleeing that destruction carried whatever sacred scrolls they could rescue out into the desert, adding them to a collection that other communities had already been contributing to for generations. What this ultimately means for our understanding of ancient Judaism, and by extension the earliest years of Christianity, is genuinely significant. The world in which these texts were produced was not one of settled, comfortable theological agreement. It was a landscape of vigorous, geographically scattered religious debate, with multiple communities each convinced they held the authentic tradition. There was no single, universally accepted version of the book of Jeremiah floating peacefully above the fray. There were multiple versions, shorter and longer and differently arranged, each one treated as fully authoritative scripture by whichever community happened to preserve it. The same pattern held true across other biblical texts represented in the scrolls, including Genesis, Psalms, and various prophetic writings. The process of canonization, of settling on one fixed, agreed-upon version of Scripture, was not something handed down complete from an earlier era. It was still actively underway, still contested, still genuinely unsettled at the exact moment these texts were being copied and preserved. And now, thanks to genetic evidence pulled from dust in a storage box, that fact had been demonstrated physically rather than simply argued theoretically. It is worth pausing here to note how differently this scientific approach performed compared to another, far more troubling chapter in Dead Sea Scroll history that unfolded around the same time. In Washington, D.C., the Museum of the Bible had spent years displaying 16 fragments purportedly belonging to the Dead Sea Scrolls collection as one of its centerpiece exhibits, pieces reportedly acquired for millions of dollars from antiquities dealers operating in a murky post-2002 market. Scholars had raised quiet doubts about their authenticity for years, but it was only through rigorous forensic material testing, chemical analysis, scanning electron microscopy, examination of ink pooling patterns, and material composition that the truth finally emerged. In 2018, testing confirmed 5 of the 16 fragments were modern forgeries. By March of 2020, an independent team of investigators confirmed that all 16 fragments were fakes, deliberately manufactured in the 20th century using old leather, quite possibly scraps from ancient Roman-era shoes, coated in a shiny amber substance designed to mimic the natural sheen of genuinely ancient parchment. It is a striking parallel. In one case, scientific analysis of the physical material behind a text revealed hidden truths about authentic ancient scripture that centuries of careful reading alone could never have uncovered. In the other, that same kind of material analysis revealed that objects long presented as sacred and ancient were, in fact, elaborate modern fabrications. Both stories point to the same underlying lesson. The material a text is written on can tell you things the words themselves never will, and sometimes those things are the only way to know whether you're looking at history or at an imitation of it. And here is where the story stops being something safely settled in 2020 and becomes something still actively unfolding today. In June of 2025, a new study published in the journal PLOS One pushed this entire field of inquiry even further, using tools that had nothing to do with genetics at all. Led by Mladen Popovic, director of the Qumran Institute at the University of Groningen, as part of a European Research Council-funded project pointedly named The Hands That Wrote the Bible, this new research combined fresh radiocarbon dating of 30 scrolls using improved contamination removal techniques at the Israel Antiquities Authority, with an entirely new artificial intelligence model built specifically for this purpose. Researchers trained that model, which they named Enoch, on detailed handwriting geometry drawn from the newly radiocarbon-dated samples. Once trained, Enoch was set loose on 135 additional manuscripts whose ages had never been reliably established. And when its predictions were checked against the judgment of expert human paleographers, the model's dating proved accurate roughly 79% of the time. Think about what that actually changes. For over 60 years, the accepted framework for dating these scrolls based on handwriting style had rested largely on a foundational paleographic study conducted back in 1961. That framework has shaped how generations of scholars understood the chronology of Jewish scripture and Jewish history throughout the Second Temple period. The 2025 study found that several scrolls are now demonstrably older than that decades-old framework assumed, in some cases by decades, and in certain instances by entire centuries. 2 specific biblical manuscripts stand out as genuinely remarkable results from this new analysis. A fragment of the book of Daniel, catalogued as 4Q114, and a fragment of Ecclesiastes, catalogued as 4Q109, were both found to have been physically written down almost within the same generation that these texts were originally composed, rather than copied decades or even centuries later as earlier scholarship had assumed. Read that again, because it fundamentally changes how close we now believe these physical documents sit to the original moment of their creation. This is not a copy of a copy of a copy, separated from its source text by generations of transmission. In these specific cases, the gap between composition and physical inscription may have been startlingly small. The broader implications reach even further than the dating of individual fragments. This new evidence suggests that literacy and the practice of formal scribal writing were more widespread in ancient Judea earlier than previously believed, potentially predating the Hasmonean dynasty, which earlier scholarship had generally credited with driving the spread of literacy and formal scripture production across the region. The study also found that the two major handwriting styles long used to separate different eras of scroll production, so-called Hasmonean and Herodian scripts, actually overlapped in their use, rather than following the clean, sequential timeline scholars had assumed for over half a century. And this is perhaps the most important detail of all for anyone trying to understand where this field is actually headed. The researchers behind this 2025 study have stated plainly that they have so far re-examined only around 10% of the known Dead Sea Scroll manuscripts using these new combined methods. 90% of this vast collection has not yet been run through this same rigorous process of radiocarbon dating paired with artificial intelligence analysis, which means, quite simply, that whatever revisions to history have already emerged from this small initial sample are very likely just the beginning of a much larger reassessment still to come. So, where does all of this actually leave us, sitting here in 2026, looking back at a discovery that began with a shepherd's stone shattering a clay jar nearly 80 years ago? The Dead Sea Scrolls are not, and never were, the private theological manifesto of one isolated radical sect hiding out in the desert from Roman persecution. Genetic evidence pulled from dust that had settled quietly at the bottom of storage boxes for decades has shown they are, instead, the accumulated sacred literature of ancient Israel itself, gathered piece by piece from multiple communities scattered across the region, diverse and contested and deeply human in their contradictions. And now, an entirely separate line of scientific inquiry, combining radiocarbon dating with artificial intelligence pattern recognition, is proving that even the timeline scholars believed they had settled decades ago was itself incomplete, built on a foundation that modern tools are only now able to properly test. We only know any of this today because scientists finally learned to look past the words sitting visibly on the surface of these ancient texts and started reading the deeper story hidden inside the biological and physical material that carried those words safely through 2,000 years of near total silence. A shepherd's stone opened the door in 1947. A geneticist's question about a piece of animal skin reopened it again decades later, and now an artificial intelligence model trained on ancient handwriting is prying that same door open even further, revealing gaps in our understanding that nobody even knew were there to look for. With 90% of these scrolls still waiting to be examined under this new combined approach, the honest answer to what else might be hiding inside 2,000-year-old dust is that nobody currently knows, and that may be the most unsettling part of this entire story.